Okay, hi everyone, Dr. Lindner again. What we're going to be looking at is the structure of the cells, a little bit more detail in terms of their function. So what I'd like for you to do is if you could pause here, I want you to make sure that you have this picture printed up, and that as we're going through them, you can look at it as I'm going through what some of their functions are, okay? So let's look at the plasma membrane or the cell membrane. And what I want you to remember about that is that it protects the contents of the cell. That's one major contributing component of its function. But we know that it also controls the movement. We spoke in another video about how the proteins and those integral proteins are responsible for bringing things in and out of the cell, those channels. And it's a semi-permeable phospholipid bilayer that's made up of proteins and lipids. The proteins are the gatekeeper. And remember, the outer part and the inner part of the cell has the phosphates, and that's hydrophilic. And then the inner part in between those two layers are the lipids, and that's hydrophobic, doesn't like water too much. The extracellular portion, let's just review some of the terms. So if here's the cell, outside is extracellular, and then inside is intracellular. Okay, so extracellular fluid is outside. The cytosol is the liquid in here. All right, cytoplasm is everything from the cell membrane to the nucleus. The cytosol is just the internal fluid. Okay, the cytoplasm is the contents of the cell. We're thinking all of the organelles outside of the nucleus, right? From the nucleus in to that cell membrane. Microvilli and cilia. So these are extensions that come off of the cell membrane. The microvilli are smaller, 1 to 2 micrometers, and then the cilia are 7 to 10 micrometers. When I think of microvilli, I think of two things, right? Here's point A, right? And then here's point B. And then here's point A. And then here's point B. Where's there more surface? In number 1? Or is there more surface in number 2? It looks like the same distance, the way I drew it. But if we take number 2 and we uncoil it, we stretch it out, number 2 is much, much longer, right? So there's an increase in surface area with microvilli. And I also think of microvilli as like a sponge. It's used for absorption. So we're going to see lots of microvilli throughout the GI tract or the gastrointestinal tract. Cilia, these things can whip around. They have these motile extensions, and they help to mobilize a cell. They help move it around, and they help to move material, especially within the respiratory tract. Okay, so if here is the respiratory tract, and in the respiratory tract, we have columnar cells on both sides. Okay, I'm just going to draw, let's say, one here. That's another columnar cell. It's going to have these cilia coming off of it. And then let's say here's another, and you have more cilia. And here's another one with more cilia. And then we have these goblet cells that sit here that secrete mucus. And the goblet cells are going to put mucus along the sticky surface on the cilia. So when you breathe in and you inhale air, if there's dust particles or debris, that debris is going to adhere and stick to the mucus that's on the cilia, but the cilia are going to be beating rhythmically upward to help get all that debris out. Okay? Smokers tend to paralyze those cilia and to break them down. Not a good thing. Organelles are all of those structures that I mentioned, the nucleus and endoplasmic reticulum and the smooth ER, the rough ER, the Golgi apparatus, mitochondria, lysosomes, peroxisomes. These are all organelles. They all have unique structures. They have specialized structures, and structure determines function in the cell growth, the maintenance, and the ability to reproduce. Centrioles and centrosome. Centrosome is this dense body that's near the nucleus, and it has, let's say, this is a centrosome, and then we have these spindles that look like this, the spindle apparatus. Okay, those are the centrioles. So the centrioles are paired organelles composed of these centrioles that are really specialized proteins. They're made of tubulin. So because they're small, they call them microtubules. The cytoskeleton, these microtubules actually are part of the cytoskeleton, where you have microfilaments and intermediate filaments and microtubules. They provide support. You're going to hear of these microtubules when we talk about muscles, and we talk about actin and myosin, these two contractile proteins. Ribosomes are really important. When we think of ribosomes, their primary function is in the synthesis of protein. These ribosomes are produced by the nucleolus, okay? When these ribosomes are attached to the endoplasmic reticulum, then we call them fixed ribosomes. And those fixed ribosomes attached to the endoplasmic reticulum are going to give them a very rough looking appearance. So they call it the rough ER, the rough endoplasmic reticulum. Okay. So the ribosomes are responsible for manufacturing proteins for secretion out of the cell, or they can also be used to help recycle the proteins within the cell membrane, because those proteins can become damaged and we have to repair and regenerate them. Free ribosomes, which are not the same thing as fixed ribosomes, these are found scattered throughout the cytoplasm, and these are designed to be manufactured for the cell. The proteins that they make are utilized for the cell. They're not used to ship them out of the cell. The nucleus and the nucleolus, they're not the same thing. Don't confuse them. The nucleus contains the DNA, whereas the nucleolus is the site of the ribosomes, the ribosomal RNA synthesis. That's deep to the nucleus. The nucleus is the control center. That's where all your genes are located. It contains the instructions. It's the instruction manual, the template to dictate what type of proteins need to be made. Okay. The ER, the endoplasmic reticulum, is involved in the synthesis, the modification, and the transport of materials within the cell, and it's continuous with the nucleus. So if we go back here and we look at the nucleus, you can see here it goes from like pinkish reddish to orange. This is the rough endoplasmic reticulum. This is continuous with the nucleus, or that nuclear envelope, really, to be more specific. There's a rough endoplasmic reticulum, and then there's one that looks smooth because there's no fixed ribosomes attached to it. And the function of the rough ER and the smooth ER are different. They're very, very different. So the rough ER is involved in the synthesis of proteins, glycoproteins, and even phospholipids. We know those phospholipids are needed for the cell membrane. It's a phospholipid bilayer, as are the glycoproteins and proteins in general. They make up the channels. So really, really important proteins. Now the smooth ER, let me see if I can move this a little bit. Okay, the smooth ER means there's no ribosomes attached to the membrane, so it gives it the smooth appearance. It is continuous with the rough endoplasmic reticulum, but this one is not involved in protein synthesis. It's involved with carbohydrate metabolism and the synthesis and the modification of lipids and even cholesterol. So the smooth ER is highly concentrated in the liver because the liver is what is making cholesterol. It produces it primarily in the evening, which is why most people that take cholesterol medication take it at night to block the liver from manufacturing that cholesterol. The Golgi apparatus consists of these flattened saccules that are involved in sorting out and modifying proteins. Sometimes it's called the Golgi complex or the Golgi apparatus. And the Golgi apparatus is going to take these proteins and it can move them towards the cell membrane to release its contents. Maybe it's a neurotransmitter, and it can open up and release it to outside. That's called exocytosis. Or the Golgi apparatus can actually produce a vacuole or a vesicle like a lysosome, and the proteins in there is a digestive enzyme. And this digestive enzyme, if this is the lysosome, there's lots of hydrogens in there. So we'll put H plus for hydrogen. And remember, the more hydrogens, the more acidic something is. And the more hydroxide, then the more alkaline or basic something is. So we're dumping, there's lots of hydrogens here. And when it finds some sort of debris, this is going to open up, releasing and dumping the hydrogen outside to whatever it's trying to destroy. And when that something it's trying to destroy becomes too acidic, it's going to die. So that's what the lysosomes do. The peroxisomes are found within the liver. We know the liver is involved in detoxifying harmful substances like alcohol and drugs. It's designed to neutralize dangerous free radicals. Peroxisomes are also involved with breaking down fat. We call that beta oxidation. When we look at fat, it's just a long carbon chain, right? Because it's a organic acid, organic compound, I should say. And whenever you split at the second carbon, they call that, right? This is alpha, then this is beta. So they call it beta oxidation, is every time it breaks the bond at every second carbon. That's beta oxidation. Okay, the mitochondria is the site of ATP production. Remember, the energy currency of the body is adenosine triphosphate, ATP. When you have adenosine and you phosphorylate it, that's adenosine monophosphate. When you phosphorylate it again, it's adenosine diphosphate 2. And when you phosphorylate it again, it's adenosine triphosphate. Okay. All right, we spoke about cytoplasm having those two components, the cytosol and the organelles. We know the cytosol is the intracellular fluid portion of the cytoplasm, and the organelles are all of the structures that we're talking about right now. No big deal. Cytoskeleton is made up of microfilaments, intermediate filaments, and here are those microtubules we saw. This is what we saw with the centrioles. The centrioles are going to be involved in cell reproduction. We call it mitosis, not meiosis, but mitosis. Okay. Meiosis and mitosis are not the same thing. So we've seen these and when we talk about mitosis, there are different phases. I call it PMAT, prophase, metaphase, anaphase, and telophase. Just be familiar with the terms. We're not going to go into too much detail with it, but it's cellular division, and I'll show you what it looks like. But prophase, metaphase, anaphase, and telophase all belong to mitosis. And those centrioles, these microtubules, are integral in that process. Microfilaments are the smallest, intermediate filaments, and then you have the largest, which are microtubules. You'll see these microtubules, again, when we discuss muscles and we talk about actin and myosin. So here are those centrioles once again. That's what they look like. They're paired. Sometimes they come in 2s or 3s like this. They call them triplets, but they're involved in mitosis. Here's the cilia and flagella that we spoke about a little bit earlier. You can see coming off of the cell membrane to help mobilize and move the cell. Flagella, this is different. The flagellum, we see that longer tail. We typically see flagellum associated with sperm. Here's the head of the sperm, and all along the body of it, there's going to be lots of mitochondria because it's energy that's needed to whip around the flagellum of the sperm. When we talk about ribosomes, we are talking about protein synthesis. Remember that, protein synthesis. Here is the difference between the rough endoplasmic reticulum and the smooth. The rough ER is responsible primarily for protein synthesis that's needed to ship outside of the cell or for the cell membrane, whereas the smooth ER, we're thinking primarily carbohydrate metabolism and fat synthesis and lipid synthesis. Here's the Golgi complex, or the Golgi apparatus. You can see it's modifying the proteins. And when it produces or releases the secretory vesicles, sometimes these secretory vesicles can move towards the end of the cell membrane. So here's the Golgi apparatus. Here's the rough ER. Okay, the rough ER created this transport vesicle. It moved to the Golgi complex, and now it's going to modify it, and it's going to repackage it. And it can send it out this way here as number 7 as a secretory vessel. So it can release these proteins outside of the cell. We call it exocytosis. It can be sent as number 8 to the membrane, and then the entire structure is used to recycle the membrane. So it's merging with the plasma membrane to help recycle it. Or it can also produce these lysosomes. Remember, lysosomes are these vesicles here that contain lots of hydrogen. The more hydrogen, the more acidic, and it's involved in removing cellular debris. So here's the lysosome. These are digestive enzymes. Remember, lots of hydrogen, so it's quite acidic. It just opens up and releases these to help remove debris from the body. Now, there are certain lysosomal disorders that can happen. There's a genetic disorder, and this genetic disorder called Tay-Sachs disease is a genetic disorder. Now, what typically happens around neurons and axons is that there's lots of lipids and cholesterol and fat that accumulate around the neurology, but we have these lysosomal enzymes that try and degrade and break some of that down. But with Tay-Sachs, there's this glycolipid that is wrapping around these structures, and these glycolipids are accumulating without the digestive enzyme or the lysosomal enzyme that's needed to try and break some of them down. So the enzyme that normally breaks down the glycolipids that are commonly found around nerve cells, it's not really there. So the glycolipids accumulate, and when they accumulate without being broken down properly, the nerve cells lose their functionality. And thank goodness, we do have genetic testing to try and pick this up, but otherwise we see seizures in these individuals, muscle rigidity, blindness, dementia, and sadly, these children die before the age of 5. The peroxisomes, these are the structures that are similar to the lysosomes. They're a little bit smaller, and they contain enzymes that utilize oxygen to oxidize and to break down organic substances, these toxins and alcohol. So we'll find these peroxisomes around the liver, even the kidneys in some degree. Now, proteasomes, these are structures that destroy damaged proteins or faulty proteins by cutting them into smaller and smaller pieces. Those are called proteasomes. The mitochondria we know as the energy factory or the powerhouse of the cell. It's a double membraned organelle. The central part of it is referred to as the matrix, and then you have these inner foldings called the cristae. And within this area of the matrix and the cristae, this is where we have the Krebs cycle taking place and the electron transport chain. And that's why it generates ATP, and that's why it's the powerhouse of the cell. These mitochondria have their own DNA. They do have the ability of replicating. We call it mitochondrial biogenesis. The more exercise you do, the more mitochondria are produced. If you're a little bit more of a couch potato and you don't do much movement and exercise, the mitochondria break down. You don't need as much. So mitochondrial biogenesis is a great thing. More mitochondria, more energy. Now, the ATP production happens when you have this 6-carbon sugar, right? 1, 2, 3, 4, 5, 6. That's called glucose. And then glucose is split. It's broken down like this into these 2 3-carbon structures. 1, 2, 3. 1, 2, 3, right? All we did is break down this bond, so you have this happening. That's called pyruvate or pyruvic acid. This step is called glycolysis, and that takes place in the cytosol or in the cytoplasm. It doesn't take place in the mitochondria. Now, once you have pyruvic acid, as long as you have oxygen available, this pyruvic acid can enter the mitochondria for the Krebs cycle. If there isn't oxygen available, then pyruvic acid goes into something called lactic acid. Okay? It's another type of sugar. When you rest and regenerate and repair, it allows the lactic acid to be converted back to pyruvate and pyruvate back to glucose. We call it the Cori cycle. And then you have energy all over again. Okay, but glycolysis takes place in the cytoplasm. The Krebs cycle and the electron transport chain take place in the mitochondria as long as oxygen is available. Now, the mitochondria, one of the theories is that it's this symbiotic relationship that took place where bacteria, first in the beginning, there was more plants. And plants go through photosynthesis and they create oxygen for us to breathe. And bacteria has its own DNA. It's so simple, it evolved quicker into aerobic bacteria. And then these cells became infected by this bacteria and they started to divide. And they took the bacteria with the cells and they started to have this symbiotic or endosymbiotic type of relationship. And those bacteria are very similar to mitochondria, or mitochondria is very similar to the bacteria in terms of their size, their biochemistry, which leads us to believe, and one of the theories is when people use antibiotics, they have less energy because not only is the antibiotic destroying the bacteria, it's also destroying mitochondria. And when you destroy mitochondria, you start to impair and hurt bodily function in which those mitochondria are needed. So where are the most mitochondria needed? Muscles. Not only skeletal muscle, but cardiac muscle and the neural system. And there are certain classifications of antibiotics that end up in tendinopathies and myopathies where all of a sudden the person's walking and they just tear the Achilles tendon. So whenever take an antibiotic, you always want to make sure there's no tendinopathies or myopathies that are associated with them. Okay. So in this picture, this is showing where you have the mitochondria. This is the cell. You got the cytoplasm of the cell. Here's glucose being broken down into pyruvate. That's called glycolysis. And again, it takes place in the cytoplasm. If there's oxygen that's available, fantastic. Pyruvate enters the mitochondria for the Krebs cycle. The Krebs cycle is producing coenzymes, primarily NADH and FADH. These coenzymes are going to be utilized by the electron transport chain, the Krebs cycle. We call it, not the Krebs cycle, the electron transport chain, which happens after the citric acid cycle or Krebs cycle. So the citric acid cycle is pushing off carbon dioxide. That's what we breathe out. We breathe in oxygen. The oxygen is needed for cellular respiration. The byproduct is going to be CO2. The byproduct of the Krebs cycle are these coenzymes that are used by the electron transport chain. The electron transport chain needs iron. There's lots of iron sulfur boxes there. And it needs CoQ10, coenzyme Q10, for the electron transport chain. It explains why if someone's iron deficient anemic, why they don't have energy and they feel fatigued all the time, okay? And also if you're deficient CoQ10, same thing. So most of the mitochondria are found in the brain, they're found throughout the muscular system, and they're found in the heart. So if there are certain drug-induced nutrient depletions that are depleting this CoQ10, you can create a fibromyalgia, you can create muscle pain, you can create heart pain, you can create brain pain, right? Dementia, Alzheimer's, cardiomyopathies, heart problems. So we want to be careful of that. Here's the nucleus, right? This is the nucleus. We see that there is a nucleolus deep within, and around the nucleus is the nuclear envelope with the nuclear pores. What comes out of the nuclear pores are those ribosomes. Okay. The nucleus contains the hereditary unit. We call them genes, and the genes are arranged in chromosomes. They look like this. We'll see these chromosomes in a second when we talk about mitosis. In terms of protein synthesis, now we see the nucleus, we see the nucleolus. It's not showing the nucleolus here, but it's the nucleus, and here's the nucleus. Here's the nuclear envelope, which we see here. And then here are the nuclear pores. And what's coming out of those nuclear pores, we can see we have ribosomal RNA, we see the ribosomes, and something is taking place here. We'll see the term transcription and translation. This is involved in protein synthesis. So transcription occurs in the nucleus, and it's the process by which genetic information encoded in the DNA is copied. It's copied onto a strand of RNA, and now that's going to direct protein synthesis, okay? It's going to direct, the DNA has the code, it's got the instructions for what type of proteins to be made, and it's the ribosomes that are going to be doing that. But it has to translate that information. The translation occurs in the nucleus, and it's the process reading the messenger RNA nucleotide language, right? It's a nucleotide sequence or nucleotide language to determine the amino acid language of the newly formed protein. So we need a translator. You have two people speaking two different languages. You got a nucleotide language. We got to be able to convert that to an amino acid language to form the protein. That's called translation. You can play this, and I strongly suggest you do, to watch protein synthesis. I can't possibly draw it out. No need for me to invent the wheel, but make sure you connect it to the internet and then just watch the protein synthesis. In terms of cell division, the cell division takes place in several phases. What you heard me mention before is mitosis. You see that here at the bottom. And mitosis has different phases: prophase, metaphase, anaphase, and telophase. You see that there's a phase called G1 and then G2, and between the two is interphase. All right. The G1 phase, the cell is metabolically active. It duplicates organelles and the cytosol components, the centrosome starts to replicate. In the G2 phase, the cell growth starts to continue. You have enzymes and other proteins that are synthesized, okay? In the interphase or S phase, that's where the DNA is replicated. That takes about 8 hours. So you've got 8 to 10 hours here in G1. You've got about 8 hours in interphase. You got about 4 to 6 hours in G1. And then you have this: prophase, metaphase, anaphase, and telophase. Okay, and there is a short video just showing these steps that we're going to show. So mitosis occurs when the nucleus of the cell divides, and mitosis results in the distribution of two sets of chromosomes into two separate nuclei. And these are the 4 phases that require. It's called PMET, prophase, metaphase, anaphase, and telophase. So in the first phase, in prophase, the chromatin is going to condense into chromosomes, and the nuclear membrane disappears, and the centrosomes start to move to their opposite poles, to their opposite sides. And we see that happening here. During metaphase, phase 2, metaphase, the centromeres of the chromosomes, they line up right here in the middle. So M in metaphase with middle, M and M. Okay, we can see that here. It's pretty cool, right? See these spindles? These are all those centrioles. These are those microtubules. And then what happens is anaphase, the centrosomes of the chromosomes, they split, and the sister chromatids move towards the opposite poles. So now they start to pull towards those opposite poles, right? It's like they throw out the fishing line. Now they're starting to reel in those chromosomes. And now in telophase, what you see is that now the mitotic spindles, those microtubules, start to dissolve, and the chromosomes regain the chromatin appearance, and the new nuclear envelope forms. And you start to see this cleavage take place on both sides. And what will happen is that that cleavage is going to pinch like this and eventually separate. And that's what cytokinesis is, right? The cleavage furrow forms, and eventually the cytoplasm of the parent cell fully splits. And when you see that split, when it's complete, then interphase begins. Okay. Now, the destiny of these cells are three different options. They're either going to remain alive and then they function without dividing, or they grow and divide, or they die. When the diversity of cells, right? Cells become tissues. They can become an epithelial cell. There's epithelial tissue. It could be muscle cells to muscle tissue. It could be into red blood cells. It could be nerve cells. It could be sperm cells. My gosh, there's like over 70 billion cells throughout the body. It's unbelievable. And then as we age, the cells gradually start to deteriorate in their ability to function normally and in the ability to respond to different types of stresses. And the number of cells that we have in our body starts to decrease. And then we start to lose the integrity of the extracellular components of our tissues. That's just normal physiology. And there's things we could do to slow them down. Obviously, the more active we are, the more active our brain is, the more movement of the human body, right? The body needs three things. It needs oxygen, it needs glucose, and it needs stimulation or movement to really drive and thrive and develop.